I am privileged to introduce our first keynote speaker of day 1, Dr. Satyendra Singh sir. He is Assistant Professor and Head of Department of Computational Biology and Bioinformatics in Sam Higginbotham University of Agriculture, Technology and Sciences, formerly AAIDU. His area of specialization, bioinformatics, genome analysis and drug designing, genome analysis, molecular dynamics, agri-informatics and chemi-informatics. Dr. Singh has done his MTech in bioinformatics from IIT Allahabad. He has completed his Doctor of Philosophy in Bioinformatics in 2013 from SHAITS. He has research and teaching experience of more than 10 years and have 36 publications so far. He has guided 21 theses at postgraduate level. He has attended 20 conferences, workshops, symposia, seminars and so on. His main area of research includes genome analysis, as I have previously explained. He is also a member of technical societies like Bio Research Society, Trivandrum, India, Society of Bioinformatics, SHAITS, Allahabad, and Italo-Latin American Society of Ethnomedicine, Italy. Let's welcome our speaker, Dr. Satyendra Singh sir, to kindly address the forum and continue with the keynote address. Dr. Satyendra Singh sir.
Good morning to all of you. I'm Dr. Satyendra Singh. I am very thankful to the organizers for inviting me to give this keynote lecture. I'm thankful to Registrar, Principal, Ma'am, Chief Patron, Convener, Mrs. Pragya Goyal Ma'am, Coordinator, Dr. Sultana Razia Ma'am, and the whole organizing committee for organizing this and inviting me for this lecture. So I request, please share the PPTs. Okay, thank you, ma'am. So the title of this talk is Drug Designing Using Computational Approaches in Post-Genomic Era. Is I'm audible?
Yes, sir, you're audible.
Okay, thank you. So as you know that in bioinformatics, there are various applications in different fields, like in drug designing, in agriculture, in environmental sciences, in nutritional science, in nutrigenomics. There are various applications of bioinformatics. But for today, we are going to discuss the specific applications of bioinformatics in the area of drug designing, especially in post-genomic era. So as this is a Institute of Biotechnology, so with the advent of modern technologies in biotechnology, we have large number of genomes that are available. Now, in drug designing, we can use the genomic information from different genomes. For example, like we can have the genes from human genomes. We can identify the drug targets. Then in post-genomic era, we can have genomes of various pathogens, microbes that are responsible for different disease. So we can analyze human genome, microbial genome, and also there are various medicinal plants. So and these medicinal plants, the genome of these plants are sequenced. So we can use the information from different genomes of medicinal plants. So we are going to discuss that how in this post-genomic era, we can use this genomic information for drug designing. Next slide, please. Right. So in drug discovery, there are different processes where we can use bioinformatics approaches. In case of computer-aided drug designing, we have two strategies. One is structure-based drug designing and the ligand-based drug designing. In structure-based drug designing, we have de novo ligand designing and the virtual screening. And for ligand-based drug designing, we have two approaches like QSAR analysis and Pharmacophore modeling. So this is basically based on when we know the structure of drug target. When the structure of drug target is known, then we go for structure-based drug designing. And if we don't know the structural information of drug target, then we go for the ligand-based drug designing. Next slide, please. Right. So from this, we can see that what are different steps that involves in drug designing. So the first is to identification of a disease. Once a disease is identified, then we go for uh, to identify the responsible drug target. Once a drug target is identified, then we go for uh, identifying the ligand or the molecule which can interact, which can either agonist or antagonist, which can interact with the drug target. Then we go for preclinical trials. Then after preclinical trials approvals, then we go for formulations, means what type of formulation will be required for that particular drug. And then we go for human trials and FDA approvals. So this is the general approach, uh, or we can say the traditional approach. Now this approach takes too much time. That's why with the advent of biotechnology, bioinformatics, and especially the pharmacogenomics, we can reduce the steps, and also we can reduce the time that traditionally it takes. Next slide, please. Okay, like from this we can see that how much time and money is involved in this drug designing process. So once a drug target is identified, then for that particular drug target, we identify the hits. Hits are those small molecules that can just interact with the drug targets. Now, just overview of drug targets. Now, what are the drug targets? Mostly, drug targets are proteins, enzymes, and receptors in our body. Drug targets, these DNA molecules are also drug targets, but in most of the cases, either the drug targets are proteins, enzymes, or the receptors. So once these drug targets are identified, then we have to find the hits that can bind with the drug target. So out of thousands and lakhs of hits we identify. Hello. Okay. Next slide, please. So right. Okay. Now this is the technological impact of on the process of drug designing. Here you can see from the very first step, means identification of disease, we have different biotechnology and bioinformatics approaches. Like for the identification of drug target, we use different approaches like pharmacogenomics, proteomics, and transcriptomics. So all this helps us in precise identification of drug target. So now when we have precise drug target, that will reduce the toxicity and enhance the potency of the drug uh, in the body. Now, once a proper drug target is identified, then drug target is isolated, and then we have to go for screening. Means what are the molecules that can interact with the drug target? For this, we use different approach of combinatorial chemistry and chemoinformatics. Chemoinformatics is just like bioinformatics, where we try to identify the chemical molecules which can be converted into a drug-like molecule. Now, in chemoinformatics, we use this approach like virtual screening. In virtual screening, we identify the molecules which can interact with the drug targets. For this, we take a different softwares uh, and the servers. And also here, we try to find out those molecules which are non-toxic or have a better absorption, distribution, metabolism, and excretion. So then we go for the preclinical testings. Next slide, please. right. Uh, okay. Here, once this identification of molecules takes place, for this we use the screening, like virtual screening, and then we go for lead generation and lead optimization. Here, once a drug molecule is identified, then we try to either remove or to add those functional groups which are essential for the better ADME properties, means absorption, distribution, metabolism, excretion, and toxicity. Next slide, please. Okay. Uh, Now, in the drug development, we use animal testing, clinical trials, and then there are various regulatory requirements that we have to follow. And once we are lucky or we are successful in getting the drug, then we can go for the patenting of the drug. Next slide, please. So lead generation. In lead generation, for the drug target, uh, lead is a minimum small compound that can interact with the molecule. So we try to identify such molecule which have a better potency and which can have minimum toxicity, and we can add those groups so that we can improve the potency. Next slide, please. Uh, then lead optimization. So when a lead has been identified, the next stage is to find the compound that are similar to it, which might bind even better. So here the main point is the binding affinity. So the compound should have a proper binding affinity with the drug target. Next slide, please. Okay. So as we were discussing that about the drug targets. So most of the molecules, they are proteins in nature. In drug targets, like we have enzymes. Enzymes, they may be inhibitors. Then we have receptors. There are various receptors in our body, like agonist and antagonist. Here, agonist means those molecules which facilitate the process, and antagonist which oppose a normal process. Then there are the drug targets like ion channels. There are different ion channels in our body. Is I am audible? Hello? Hello?
Yes, sir, you are audible.
Okay, okay. There are different ion channels in our body, like sodium potassium ion channels. These ion channels perform very important functions. So these many times these ion channels are our drug targets. Then there are different transporters which transport the drugs, help in the distribution, and our DNA is also a drug target. Next slide, please. Okay. So failure of compounds in development. Most of the time, the molecules failed because of poor ADMET properties, means absorption, distribution, metabolism, excretion, and toxicity. Then next, around 29% of compounds fails due to lack of efficacy. Then around 21% fails due to their toxic nature, and around 6% due to some market agents. Next slide, please. Now, the compounds should have drug-like properties, and only those compounds which have drug-like properties, they can be converted into a drug molecules. So compounds, in order to be drug-like compound, should follow some rules. So these are the some rules, like empirical rules. One of most important criteria is Lipinski rule, known as Lipinski rule of 5. So in Lipinski rule of 5, we check about molecular weight, partition coefficient, hydrogen bond donor, and hydrogen bond acceptors. So all these parameters are checked, for a compound to be a drug-like molecule. Next slide, please. Okay. So these are the rules. This is most important, that a compound should have molecular weight less than this 500. This hydrogen bond donor acceptor should be less than or equal to 10. So this is the common strategy like we use in drug designing. We check the binding of ligand and the receptors. Are they are proper binding, so that a ligand can perform its function. Next slide, please. Okay, so as we discussed, this is two strategies: structure-based drug designing and the ligand-based drug designing. So when we know the structure of drug target, then this is structure-based, and when we don't know the structure information of drug target, then this is the ligand-based. For structure-based, we use de novo drug designing and the virtual screening. So as in structure-based drug designing, as we say, we have the structural information of drug target either through the X-ray, NMR, or through the homology modeling. Next slide, please. Okay, so this we use when we have the structural information of drug target. So drug target and the ligand molecule, they can bind or they can fit into the cavity or the binding site. Next slide, please. Okay, and one approach is this, that when we don't know the structure of drug target, then we can use the computational approaches. Here bioinformatics comes into play. We have this method like comparative modeling or homology modeling. So by using this homology modeling, directly from the sequence of a protein, from primary structure of protein, we can find out a tertiary or quaternary structure of protein, and we can validate the structure of protein by using different softwares, different servers. One of the most important is Ramachandran plot, then ProCheck, WhatCheck, Verify 3D. These are the different softwares with the help of which we can validate the protein structure, and then we go for the drug designing. Next slide. So we use different softwares like we have Lig Builder, LUD, and this homology modeling, structure-based drug designing that are used in the de novo drug design. Next slide, please. Then the virtual screening. This is a component of chemoinformatics where thousands and lakhs of compounds can be screened by using databases in bioinformatics like Zinc database and PubChem database. So we can explore this database to identify the compounds which can interact with our molecule and the drug target. Next slide, please. Okay. This is a very important topic in bioinformatics that is known as docking. So molecular docking is a study of how two or more molecular structures, for example, drug and enzymes or receptor of protein, fit or interact together in 3D space. Searching and scoring are the two components that typically make up a computational docking. So docking is a process where we take a compound, a chemical compound, and we try to see its binding with the drug target. And out of thousands of compounds, we select those compounds which are having good scoring and better affinity with the drug target. Next slide, please. Okay, so this is example that how this a compound is docked and it is interacting with the binding site or the active site of the drug target. Next slide, please. Okay, this is that searching and scoring means out of thousands of compounds, we search only those compounds which have proper binding affinity and proper score with the drug target. Next slide, please. Okay. So there are the different softwares with the help of which we can do the docking. This is a name of the softwares are Dock, Autodock, Flex, and Hex. Next slide, please. Now this is a ligand-based drug designing. In ligand-based drug designing, we don't have structure information of drug target. So then we go for QSAR analysis, quantitative structure analysis, and the pharmacophore modeling. Next slide, please. Okay. So from this, you can see that out of various compounds, we try to find out a pharmacophore. Pharmacophore is a pattern which is generated by overlapping and superimposing different compounds so that we have at least minimum information that which type of compound can bind with the drug target. Next slide, please. Okay, so this is a QSAR where it is based on that when we change the structure of a compound, then its biological activity is also changed. So we try to keep the balance between what activity we require and how much structural changes we can make into the compound. Next slide, please. Okay. So here we use different mathematical approaches. Like we take a set of compounds, we check their activity, then we devise their molecular descriptors. Now we keep on changing and modifying the molecular descriptors until we achieve the desired activity. Next slide, please. Okay, so this is you can see that how from a lead compound we can generate the pharmacophore model. Next slide, please. Right. So this is hello. Is I'm audible?
Yes, sir.
Okay. Okay. Thank you. So this pharmacophores modeling involves the essential substructural moieties of a molecule necessary for its pharmacological activity are known as the pharmacophores. Compounds having similar pharmacophoric groups often have similar biological or pharmacological activity. Next slide, please. Okay, so this is how from a pattern of different molecules, we extract the information that what type of a pharmacophore can we develop that can be converted into a lead compound. Next slide, please. Okay. So there are various successful stories by using CAD or computer-aided drug designing. So you can see that a number of drugs that are designed by using this computer-aided drug designing. Okay,